begin. Now here, let's talk about the human trypanosomiasis, which is the disease. So we have already discussed about the basic part of the disease section, which are how the structures are and also what are the different components of it. Now we'll be talking about its different disease that are caused by this particular trypanosoma. Now the disease caused by trypanosoma can be divided into two different sections. One is the disease called the disease actually called sleeping sickness, but it is a kind of African sleeping sickness, and the second type of disease is called American sleeping sickness. So let's try to talk about it. It is African trypanosomiasis or African sleeping sickness, and in this case it is actually started in West Africa by the Trypanosoma gambians or Trypanosoma brucei and we already talked about that. Now it sometimes can also transfer in East Africa too, because it can be found in a whole lot of African regions. In East Africa it is caused by Trypanosoma brucei rhodesiensis. Okay, and for the American trypanosomiasis, it can be caused by Trypanosoma cruzi. Right now, usually American people can get this disease if they visited Africa in some region. Otherwise, they won't. There is no way to get this disease because actually, this Trypanosoma or this tsetse fly, which are the carrier for this or the vector for this disease, is not present. Is not found in America. It is mostly found in Africa, right? So if an American people move to Africa and if it is being infected and bitten by those tsetse fly, infected tsetse fly, then there is a chance of getting the disease. Now if we look at the trypanosomiasis in Africa, what we can see, the vector is tsetse fly. Look at the spelling, look at the pronunciation again, tsetse fly. And Trypanosoma brucei gambians is the agent for this disease in West Africa, and Trypanosoma brucei rhodesiensis is the example of for the East African disease. Now in this case you can see the disease caused by brucei gambians causes chronic infection lasting years and affect different countries of Western as well as Central Africa. Okay, now in case of brucei rhodesiensis, it causes acute illness which is much more worse than the disease caused by gambians, and it is lasting several weeks in southern and eastern Africa. So though this brucei rhodesiensis or Trypanosoma rhodesiensis cause a very small duration of the disease, almost 1 or 2 or 3 week for the disease, but this disease symptoms are much worse. But gambians can cause the disease more often throughout the Africa. It is very long lasting, like years, but it is not that much harmful. Okay, now how this disease is transmitted. Now remember, I've talked about this vector. Talked about the vector, and the vector is called as tsetse fly, and this is the structure of the tsetse fly here, right? It is found in rural Africa. There are over 22 types of this fly, and all strictly feed on blood. So they feed on blood. This is very important. Feed on blood, otherwise this disease may not be happen, right? Now they usually breed on rivers and streams, and after that they can come and infect other individuals. They can eat and feed on blood and then can spread this trypanosoma from their salivary gland to the bloodstream of human being, right? And then the disease can be spread. Now a pregnant woman could pass this disease to the child, and the child become infected due to this kind of situation. So this can be congenital in such a way, right? So if we look at the African map, we can see this whole red region like Uganda, Kenya, Tanzania, Malawi, Ethiopia, and this whole section of this Middle Africa is almost in throughout the Middle Africa we can see tsetse fly, and also North and East Africa also, right? So this is a kind of tough situation throughout the Africa because tsetse fly lives there. And here comes the different stages of this disease, right? So here in this case, let us first know this symptoms, the signs, that I means infective stage, D means diagnostic stage. Okay, so let's talk about it. So the first stage is that, let's say the tsetse fly takes a blood meal of a man. Now the tsetse fly here we are talking about, the tsetse fly here is infected. It is infected with or say it is infected with trypanosoma. So it is carrying trypanosoma. It is carrying trypanosoma. Now as it is carrying trypanosoma, if it infects, if it if it is feeding onto the blood of human being, it will inject this trypanosoma inside the bloodstream of this individual, right? So it injects it. After the injection, the injected metacyclic it is called actually trypanomastigotes. Once it is ingested, once it is not being developed and fully adult yet, it is called as trypanos trypan try trypomastigotes, sorry for that, trypomastigotes. Now this is called the metacyclic trypomastigotes, and this metacyclic trypomastigotes transform into the bloodstream into trypomastigotes, simple trypomastigotes inside the bloodstream. Now these trypomastigotes multiply by binary fission, as you can see in this picture. The binary fission means, remember, if this is a cell, it will make a pinch like that, and then it will divide into two cells. So it's a kind of simple division like bacterial cell division. So it multiply by binary fission in various bloody fluids like blood, lymph, and spinal fluid also. Now usually they multiply in this blood and lymph nodes, and they stored in the lymph node, and they are moving through blood and lymph vessels. Okay, after that it will move to blood, and then it is called the trypomastigotes in the blood, so you simply know as that. Now still this part, till that part, we are only talking about the diagnostic phase. Now if we take blood from this individual and look for it, we can find this trypomastigotes in the blood of the infected individual, right? After this stage, this trypomastigotes will go and freely moving around the bloodstream of human beings. So this stage can last for years. This can last for several months to years. So they can just go on moving on, and they will provide some milder symptoms. So we can see, if you look at the graph of the symptoms, we'll be looking at in the later slides, that we can find a kind of rise in the infectivity, a kind of symptoms, and a fall, then again a rise and fall, again a rise and fall. So we are going to see a lot of infections like that. Now the infections usually, getting the graph like that, is called kind of persistent infections, because it is not going away. It is slightly it is sometimes getting very high, then again antibodies are raised by the body, antibodies are killing some of the pathogens, and again the infection is going down. But after certain time, it will vary their antigen in such a way. Due to this antigenic variation, they again start to fight against the body immunity, and they again start to rise, but again new antibodies will fight against them and taking them and killing them down again, right? So this is a kind of system, right? Now here what we can see, that this trypanosome or trypomastigotes, they are moving around the bloodstream, and in those case, suppose another tsetse fly moves away and moves in, and it will start taking another blood meal from this individual. Now in this case, individual is infected, but the tsetse fly is uninfected. But after this bloody meal, what they can get this trypanosome at their salivary gland. Now at the salivary gland, so from throughout this salivary gland, what we can see, the bloodstream trypomastigotes transformed into procyclic trypomastigotes in tsetse flies midgut. So it will move on through the salivary gland, through the uptake region, through the midgut of this tsetse fly, and it will be called as procyclic trypomastigotes. Now here they will start to divide again using binary fission. After certain divisions, what it will move, these procyclic trypomastigotes leave the midgut and transformed into epimastigotes. Now once these epimastigotes are made, they again start to grow and divide again. After the division, these epimastigotes multiply in salivary gland, and when they come to the salivary gland, they transformed into metacyclic trypomastigotes. Now these metacyclic trypomastigotes again move, and they are stored in the salivary gland of the tsetse fly, so that now second time, if the tsetse fly infect a healthy individual, this cycle repeats again and again and again. Now you can see the multiplication can be found in the simple trypomastigotes in the bloodstream of human being. It can be found in the midgut of tsetse fly, and also you can find this division going on in the salivary gland of uh, fly, tsetse fly. So these are the different stages. You can see most of these dangerous kind of stages, they're growing in both this case. The growing and division are going on most of the time in the tsetse fly body. So if the tsetse fly is carrying this trypanosoma, there is no way that the individual won't get it. They will surely get it, and after certain time they will develop into sleeping sickness disease. Okay. Now, if we look at the insect stage in the bloodstream, what we can get that the different stages express different sets of surface proteins. Remember, we have already talked about the surface proteins. Varying the surface proteins, they can vary the response of immunity, and that provides them some time for their growth and development. Right now, the insect forms have larvae mitochondria with many cysti. It's not that much because they require energy, they require for their movement and all these things. So that's why they require more amount or more region or more space inside their mitochondria. That's why they are having more cysti. Okay, more cristae, not cysti, cristae. And insect stages have an aerobic metabolism and a full respiratory chain. So that's why they are not anaerobic. They are aerobic. That's why they are living in the bloodstream and midgut. So that's the reason that they are not staying in the Setsifly midgut for longer period of time, because they won't uh, get enough amount of oxygen there. So that's why they start to come out to the body's circulation systems like blood or lymphatic system, or sometimes in salivary glands for the septic life, right? Now, bloodstream forms only engage in the glycolysis and excrete pyruvate and glycerol outside, right? Note that transmission stages do not replicate and are arrested in development, right? So that's a very important point. So until and unless they are mate with some uh, female trypanosome, they won't go and replicate. That's the very important part. Okay, so that's the kind of transmission stage. Transmission stage means these are the stage when they are moving from the tsetse fly to human, or from human to tsetse fly, right? Now, if we look at the stages in human body, because we're interested in how it is going on inside humans, so if this is the injection of this trypanosome by the tsetse fly, and first it made to this blood vessel, and through this blood vessel or lymphatic, they will move into the secondary stage, where they can move from blood vessel to other tissues also. I haven't mentioned it, but still they can migrate to other tissues because they can move using their flagella. But after that, they can sometimes, the third stage and the most dangerous stage can occur. Sometimes it occurs, sometimes it does not. But if it occurs, it can migrate through, it can cross the blood brain barrier, and it can move to the to the brain region, and it will start damaging the central nervous system. That's the dangerous, because CNS, they start damaging CNS sometimes. That's dangerous, right? So usually the why they are kind of dangerous, because they can avoid our immune system. So if we talk about their virulence and pathogenesis, so they can create pathogenic factors because they are dangerous, they are having all these things. But the most dangerous thing about trypanosomal infection is that they can avoid the immune system by antigenic variation, right? So they can change their outer membrane coat in certain case, the protein produced in certain case, so that they can fool our immune system.